Dear all, dear participants, of course, I first would like to thank the organizers for having invited me on this interesting conference. Um, the question I got is to tell something about novel insights into platelet signaling and phenotyping. Of course, platelet signaling and phenotyping is kind of are complex matters, so I need to focus on specific points. But I would like to do that in a way that hopefully everybody of you can take profit of this presentation. So I will tell something about thrombin and collagen receptors and platelets and what's happening downstream of the receptors, also new insights, how those receptors are working and how those receptors are activated. And also I will actually start to introduce some useful tools that you may know or may not know, which are very helpful in understanding the complexity of genes, transcripts, and proteins related to platelet function. As a first, an overview, general overview of how we think now in these days that the platelet signaling system works in the context of thrombus formation. What you can see on this slide is that after rupture of an atherosclerotic plaque or after denudation of the endothelial, collagen fibers become exposed. Fibrillable factor will bind to those collagen fibers. Platelets under high shear conditions will bind to that fibrillable factor, will be slowed down, and as a consequence, platelet collagen receptors will interact with the collagen that is underneath the fibrillable factor, and this collagen receptors then cause an activation signal, and the activation signal leads to shape change, subsequently integrin activation, secretion, and platelet proconductivity. And what you can see in the heat bar is that depending on the extent of the calcium signal, different responses to the platelets will take place. Important point is their secretion. Secretion includes secretion of autocoids like thromboxane and ATP, and these autocoids will be released in the blood, will activate other platelets, other flowing platelets, and as a consequence, the platelets will aggregate, form together by means of integrin fibrinogen bridges, and so you gradually get the buildup of a platelet aggregate of a thrombus. Some platelets, those platelets that are very high in calcium, and actually these are the platelets that are heavily stimulated via GP6, escape from this route, and those platelets become procoagulant. They expose the negatively charged phospholipid phosphatidylserine. Coagulation factors bind to those platelets, and as a consequence, the thrombin is formed, and then we will detail it later. The thrombin activates then so-called G protein coupled receptors that enforces the stimulation of the aggregate, enforces the platelet activation there, and in addition, thrombin makes fibrin, and fibrin is there to consolidate the clot. And 15 years ago, we thought that this was the end stage, but I will come back later in my talk to see that is quite a bit more happening even after fibrin is formed. Before going on, I would like to give some idea of, let's say, the complexity of the platelet signaling part. What you can see on this slide is that if you look at the human platelet transcriptome, and quite recently we could establish the complete human platelet transcriptome, you see there is almost 15,000 protein encoding transcripts there, and of which over 5,000 proteins have recently been identified, of which many over 1,000 signaling proteins. So the few signaling proteins I will introduce to in more detail later on is only a fraction of the total number of proteins that we know, and you can appreciate there are hundreds of membrane receptors and channels, but the hosts hundreds of protein kinases and phosphatases, and many other signaling and adapter proteins that probably all in some way contribute to either platelet formation or platelet activation. And to increase the complexity, if we compare the identified proteins so far and the non-identified proteins so far, you can appreciate that especially with the signaling proteins, the majority of transcripted and code for proteins, the corresponding proteins have not been identified. So at present, we don't have a clue of what these are doing. Nevertheless, we know that some of those signaling proteins are important, and we know that because there have patients been identified that miss those proteins, and transgenic mouse have been made that miss those proteins, and we know from patients and in mouse studies that the effects is on thrombosis and hemostasis. So before introducing some of the specific signaling pathways, I would like to present some publicly available websites and databases. These I will introduce you a little bit about the GeneCart, UniProt, Reactome, Omen, and the Blueprint Blood Atlas. So if we start with GeneCarts, that is a composite database of human genes and products that has been composed by the Weizmann Institute of Science, and it's a very useful database because it combines information from various other databases as well, and is also quite efficiently updated. So if new genes have been identified, then you will see that within a few weeks, those will be introduced in GeneCarts as well. So what does GeneCarts offer? It offers insight about human gene function, location, and structure. And the slide here gives an example. If I select the gene GP6 for the main collagen receptor on platelets, GeneCarts gives information on promoters and enhancers of those genes. It gives direct links to other databases. It gives information on protein products, on splice forms of the protein products, and also variants, proteins present there, of very useful information about post-translational modifications. And what is also quite useful, I think, it gives also lists of antibodies related to the genes for research and protein products that are available for research. You get information on animal clones, on inhibitory RNAs, on pathways, interactions, on drug products that are known, transcripts, and even the expression profile of the gene in the human body is indicated. And there's an extensive list of database sources and so on. That altogether makes this an easy accessible and quite comprehensive and complete database to have a look at. A different type of database is the UniProt database, collected by EMBL and eBay, and that's a database on proteins. And it's not only human proteins, but proteins of many other organisms as well. What's useful in this database, again, I have typed in GP6 here, then in this database also there's a reviewed part of GP6 is indicated here. So a gold star type of page, and that page gives quite useful information on the protein GP6 in platelet. So protein, gene, organism, also brief the function. It links to gene ontology. It gives information on the taxonomy, on the subcellular location of the protein, of the pathology, of the mutations known, of the pathology known, and biotech information. Extensive list of post-translation modifications. So essentially all glycosylation sites and phosphorylation sites are indicated in this on this page. Expression of the protein, interaction and similarities with other proteins. Importantly, also the three-dimensional and secondary structure and the domain structure of the protein, sequences, variants in the sequences, genome annotations, and also links to other databases. A quite different database is the Reactome database that has also been started by EBI, but now working together with several other institutes. And that is a manually curated database of protein protein interactions. So manually curated indicates that any interaction in proteins that's indicated in this database that has been looked up in the literature and that is foreseen for a reference. So from this perspective, this is an let's say better curated database than gene ontology, which gives more information, but this is really well documented, and every protein interaction you can find back in the references. If you type in a certain gene or protein or a certain list of proteins, you get this very nice overview of possible functions related to signal transduction, metabolism, disease, and so on. And then you can zoom in and then zoom in until the enzyme, and then find in which enzymes have which interactions, or which proteins have which interactions with other proteins, and where in the human body, or where in the cell function system those proteins play a role. So in summary, you find reactions and pathways. You can analyze those, you can find those. You find information on the molecules and the structures and the expression of all those proteins. There's a very nice way of network visualization there. And it is possible to upload even long lists of genes or proteins, and then find in, by looking at false discovery rates, find in which pathways are most specific and most closely together, fit most closely together with the list of proteins you have inserted. Then we move on to, again, a quite different database, the OMIM database, Online Mendelian Inheritance in Man. And that actually is a catalog of human genes and human genetic disorders. So you will find pages there of all human genes and of all disorders, all diseases where there's a genetic link there. Again, I have typed in GP6 there, and you see a list of pages where GP6 play a role in terms of diseases and the gene. So what you find here is pages per human gene, pages per human disease. Interestingly, also a history of clinical features. So when was the disease discovered? What is known over the years about the molecular genetics of that particular disease. You find short information on protein description and gene function. You also find interest on human allelic variants that link to different activity of the gene or the protein you're interested in, and there's a quite extensive reference list. The small disadvantage of this page is it's not frequently updated, and it might take a while before, for instance, a new gene that has been discovered before it also enters into this database and is updated into this database. The final database I would like to introduce is the Blueprint Blood Atlas. That is a genome-wide RNA expression atlas for actually all precursor cells, bone marrow cells, and blood cells. As you can see on the slide, it gives lists on genes, transcripts, also microRNAs, novel genes, transcripts, and also circular RNAs. And if you type in GP6 there, you quite nicely can see that actually you find that the GP6 protein is only the GP6 mRNA, messenger RNA, is only expressed in platelets and megakaryocytes, but essentially no expression in no other blood cells or precursor cells. But in contrast, if you type in the terminal receptor PAR1 gene is F2R, then you will find that it's highly expressed not only in platelets and megakaryocytes, but also on endothelial cells and T cells and K cells. So this is a genome-wide database, so actually you can type in any protein encoding RNA or pseudo RNA or RNA gene that you where you're interested in, and then find how the expression is across stem cells, hematopoietic stem cells, derived cells, and the major cells that are flowing in the blood and that are present in the vessel wall. So now, if you now move to the proteins specific receptors, as I told you, I would like to present the collagen receptors and thrombin receptors, and these actually are the three types of receptors I would like to focus on, starting with GP6, the collagen receptor, then starting with GP1B59, it has a function as a receptor for wearable factor and also for thrombin, and then the two thrombin receptors in human blood is PAR1 and PAR4. When we start at GP6, you will see here quite a complex overview of platelet signaling, but look at the very right end. There you find GP6, and that item D is an item called item receptors. You find there GP6 as a receptor for collagen, also indicating fibrinogen. You find a similar receptor there, it's called GLEC2 as a receptor for protoplannin. And then you can see with the arrows that apparently activates a number of protein kinases called SYK and SFK, indicating Src family kinases. Outcome pathways is phospholipase C gamma, that's intermensal causing calcium responses and then downstream secretion. And another type of outcome is PI3 kinase activity that leads to various other protein kinase activities, and the consequence, the end of that, is actually integrin activation. So if we detail there a little bit more in this scheme, actually that has been made 15 years ago, the GP6 signal is a cascade of tyrosine phosphorylations in the LAT signalosome. That scheme is still valid, but with a few buts. But I first will explain the scheme and then detail into the buts. GP6 tends to, for activation, GP6 leads to dimerize onto cluster. There's only 9,000 copy per platelet there. That is not, that is a few, but not, let's say, too many. The dimerization of GP6 actually, because there's a co-receptor there, it's called the Fc receptor gamma chain. So the dimerization or clustering of GP6 also leads to a dimerization of the Fc receptor gamma chain, and that Fc receptor gamma chain contains a so-called item domain. That item domain, if that two of those item domains come close together, that leads to activation of tyrosine kinases of the Src family kinases, Fin and Lin. Once that item domain is double phosphorylated, then a next tyrosine kinase comes into play, because that the double phosphorylated item binds SYK, protein kinase, tyrosine kinase SYK. That is an important protein in enforcement of the total signaling cascade, and that protein can phosphorylate many other proteins that assemble together in the so-called LAT signaling. So what you can appreciate from this slide is the LAT signaling also contains phosphorylation, activation of phospholipase C gamma, causing calcium responses and secretion, and also forms PIP3 by PI3 kinase, and that leads to the integrin activation. So it's a signaling cascade that actually has two major outcomes linked to platelet secretion and platelet integrin activation. If you now look from the back, the classical GP6 ligands are all ligands that are known to dimerize or cluster GP6. Quite a few collagen type ligands. The classical ligands include fibrillar collagen 1, HORM, is fibrillar, it's fibrous, so that fibrils can easily cluster. GP6 molecules on the same platelets. There's also native collagens like collagen type 3 and 4. Those do not find fibers and have a less clustering capacity, which still cause dimerization of GP6 molecules and hence activation. It's a collagen-related peptide that has been made, that has been artificially made, and because it's cross-linked, it has a high capacity of clustering GP6 and therefore causes high activation there. It's a snake venom convulsion that also causes clustering of GP6 and some clustering antibodies. Also indicated in this slide that the signal can be stopped, so to say, by cleavage of the extracellular domain of GP6 by ADAM10, and that gives soluble GP6, which sometimes has been used as a biomarker for in situ platelet activation. Next to the classical ligands, GP6 ligands, we know quite a few novel ligands. These include laminin, galectin-3, fibrinogen, fibrin fibers, and even diesel particles. So what you can see from this slide, it's not only collagens that can activate GP6, but it's also many other type of proteins that are present in the plasma, or proteins that are present in the clot, even fibrin, that are capable to activate GP6. So in fact, meaning that fibrin formation is not the end stage of thrombus formation, but it's a kind of intermediate there. The various drugs now in clinical trials related to GP6, Clemzocimab, which is now tested in a trial for stroke patients. Rivacept is another one, that's a GP6 protein construct, and various laboratories are now also making nanobodies and other antibodies that block the interaction of GP6 with various ligands. One of those is, one example is given on this slide. This is a slide where patients have been examined, quite rare patients with mild bleeding disorder, derived from Chile, heterozygous GP6 or homozygous GP6 deficient patients. And if you look at the thrombus formation of those patients, you will see at the left side the thrombus formation on collagen, fibrillar collagen, is reduced but not abolished. And the thrombus formation on laminin is substantially inhibited in those patients that do not that have platelets that do not express GP6. Markedly also, if you look at a parameter of the procoagulant activity, so the staining with annexin 5 that stains for exposed phosphatidylserine, you will see that that is completely abolished in all patients, in the patient both on collagen and the laminin. So from that you can see actually the procoagulant activity. That for sure is a platelet response that completely relies on GP6 activation, while the formation of thrombi or aggregates also involves other receptors in some way that can compensate for the absence of GP6. Another tool to block GP6 is this antibody, the 912 Fab antibody, that is a precursor of the antibody that's now used in clinical trials. And what you can may appreciate on this slide, that if you look at thrombus formation fibrin on the left part of collagen type 3, you will see a substantial decrease in platelet adhesion, platelet activation in terms of P-selectin expression and PS exposure in the presence of the antibody. So that this also proves that GP6 actually is active in platelet adhesion and platelet activation, not only in collagen, but also a very other type of services. That's not all, because there's also now good evidence now that GP6 can play a role in venous thrombosis. And the evidence actually comes from information collected on this slide. There's a single nucleotide variant in SNV of GP6 that is quite common, that associates with increased GP6 expression on the surface of platelets. And epidemiologists have shown that this SNP associates with an increased risk of venous thrombosis. as have been shown in very individual studies, but also in meta-analysis. And interestingly, a similar type of effect has been seen also in the single nucleotide variant of the Fc receptor gamma chain. So this suggests, we do not have a proof there, but it suggests that perhaps the interaction, plated interaction via GP6 with fibrin plays a role also in venous thrombus formation, and that increased expression of GP6 unplated by interacting with fibrin can maybe result in bigger thrombi, and that could explain the increased risk in venous thrombosis. So what's new on GP6? Actually the native GP6 collagens, the native GP6 ligands, means collagens, fibrin, laminin, are in fact only weak agonists. They cause limited receptor clustering and are co-receptors that appear to be required for the signaling pathways. There seems to be a role of GP6 not only in arterial for most studies, but also in venous thrombosis. The protein receptor CLEC2, which shared the signaling pathway with GP6, actually does not have a known ligand in normal circulation. Protoplanin is present in plaque, but not in the blood. Yet it is a role in thrombosis and inflammation. So there's another receptor next to GP6 that contributes, CLEC2, that contributes to thrombosis and hemostasis, but we still do not know the ligand there. So there's quite some additional research is needed actually to find this ligand and also find the likely co-signaling pathways that are triggered by GP6 and its kind of sister receptor CLEC2. If you now move on to the next protein, I would like to introduce here the GP1b59 complex. It's on the upper part of the slide, involved in platelet adhesion, via von Willebrand factor. Indicated on the slide is signaling via 1433 proteins and subfamily kinases. So if we zoom in a little bit here, we can see here a cartoon of the structure of this GP1b59 complex. What's indicated there is complex protein, 2 chains likely of GP1b alpha, 2 chains with small chains of GP1b beta, 2 chains of GP6, and in between there is GP5. It's much more abundant on platelets than GP6, 20 to 25,000 copies of the complex per platelet, so quite abundant there. What's indicated there here also is that the most massive protein there is the biggest protein is the GP1b alpha chain. As many so-called leucine repeats, these are the yellow circles. And there's also a negatively charged part there, and there is a part that is highly glycosylated there. So it's kind of, you could have kind of sticky protein to which many other proteins of the plasma can bind. It extends into the cytosol, and there it links to the actin cytoskeleton. What you can appreciate is that the 463 proteins in some way are needed for this interaction and to keep the protein in place. There's quite some discussion still on the extent of signaling of this GP1b59 complex. All types of cartoons have been showing less or more signaling, but I think it is fair to appreciate that there is evidence for limited signaling when this complex has bound active von Willebrand factor there, and the signaling especially is via subfamily kinases, SFKs. Some cartoons also indicate an involvement of the Fc receptor gamma chain. I wonder if that's really the case. It cannot be excluded that in fact there is kind of crosstalk with GP6 there, but it is also very clear that the limited signaling probably needs to have enforcement by secondary pathways. So most papers reporting signaling via this receptor also recognize that part of the signaling is enforced by secondary pathways like ADP and thromboxane. So if you compare the classical ligands of GP1B alpha, it's mostly von Willebrand factor. It's a different type of von Willebrand factor. It's von Willebrand factor that binds to the active GP1B59 conformation. So under high shear conditions, the complex gets a conformation change, and that facilitates interaction with von Willebrand factor. The binding is also enforced by von Willebrand factor that is activated itself by high shear or unfolding, for instance, by binding on the collagen surface. Ultra high multimeric von Willebrand factor also favors the interaction with GP1B59 complex on platelets, and also von Willebrand factor that's activated by various snake venoms like botricitin or russicitin that also favors the interaction with GP1B59 on platelets. Also the GP1B alpha domain can be cleaved by an ADAM protein, but in this case it's ADAM17, and then we get glycocalycin circulating in plasma, and that's also sometimes used as a biomarker indicating for activation of the complex in situ. But there's additional ligands for this complex as well. And there's all ligands that bind to the GP1B alpha chain. Thrombin binds to the negatively charged part. That probably enhances platelet activation. There's an indication that, but it's not very well clarified in the literature. There's still some research to do. Also, coagulation factors like factor 11 can bind to GP1B alpha. There's evidence that it enhances the coagulation, but in fact, also other coagulation factors and anticoagulation factors can bind to this complex. So you could say that the GP1B5 alpha, in fact, brings together quite a mixture of coagulation and anticoagulation factors that in some way will influence the coagulation pathway, probably via factor 11 activation, but is still more work needs to confirm that. Interestingly, GP5 can be cleaved by thrombin. There's evidence that this reduces platelet activation. The mechanism is not quite clear yet. Drugs approved is clapacizumab that binds von Willebrand factor and then inhibits interaction with GP1B alpha. That's approved for the use in TTP. And there is also a series of new anti-von Willebrand factor and anti-GP1B antibodies present and being developed that also may be used in the future for similar approaches. This slide gives an indication of an antibody, actually the REC-35 antibody that was generated already many years ago. There's an antibody that blocks the interaction of von Willebrand factor with GP1B alpha, and that you can appreciate there that at a collagen type 3 surface at the left side and a fibrin surface at the right side, the presence of that antibody really blocks platelet adhesion, blocks platelet activation in terms of P-selectin expression and PS exposure, maybe even to a higher extent than the blocking of GP6. So actually, you could say in the conditions of high arterial shear stress, this interaction of GP1B59 complex with von Willebrand factor is really an important determinant of the thrombus formation. So what's new? The key roles of GP1B59 in platelet formation, thrombosis, and hemostasis. Platelet formation we know because the patients that lack GP1B59, those patients have a low platelet count and also platelets that are quite big, so condition of microthrombocytopenia. We know that's high abundant, that protein complex, and it has interactions with several correlation and anticoagulation factors, but it's incompletely understood how the low signaling acts via Src family kinases. And on the other hand, we know in terms of importance that all disease links related to GP1B59 is always related to abnormal von Willebrand factor binding. So from that, I would assume that the main function, let's say in health and disease, of the protein complex still is the interaction with von Willebrand factor. Then, when a very short comment on the last receptor type that I would like to discuss, that's at the right side again. You see at E, you see thrombin, there's two receptors indicated there. 7 transmembrane receptors called PAR1 and PAR4 activate via G proteins indicated as G alpha Q that activates again phospholipid C, goes to calcium, and is indicated G alpha 12-13 that causes shape change via the Rho kinase activation system. If we zoom in a little bit on that, in the present slide, this redundancy there in terms of what thrombin can do with platelets. So the current idea still is that at low concentration of thrombin, first the PAR1 receptor is activated and cleaved. Thrombin cleaves the receptor, neo and terminal site then activates actually the receptor and causes the signaling event. It's weak signaling via PAR1, so the completion of platelet aggregation that requires the presence of feedback via ADP and ADP receptors. But at later stage and at higher thrombin concentration, then the second receptor comes into play, this PAR4 receptor, and that, so to speak, completes the platelet activation process. So a stepwise approach there, first activating platelet PAR1 and then activate PAR4. From the platelet perspective, it's quite interesting how that works, because we know that on some platelets thrombin is formed, on propionate platelets thrombin is formed via PS exposure, and then what probably happens during the process of thrombus formation, that forms thrombin that moves to other platelets, non-coagulant platelets, and then cleaves the PAR receptors, and then we get signaling. So there's a crosstalk between platelets, some platelets forming thrombin and other platelets responding to thrombin. Also here, it's in recent years we have understood that the real picture is a little bit more complex than indicated on the previous slide. So if you look at the original PAR1 agonist, that's thrombin, mesothrombin, and TRAP6. But now in these days we also know that many other proteases like matrix metalloproteinases, kallikreins, plasmin, and trypsin-like protease also can cleave PAR1 and activate PAR1. There's 2 drugs approved, atopaxar and varopaxar, have been used in the setting of arterial thrombosis there with reasonable success. The same holds for PAR4. The original agonists were thrombin, mesothrombin, and a PAR4 peptide, but in these days we also know that various catepsins produced by platelets, kallikreins, and also plasmin can also cleave PAR4. Importantly here is that several of the proteases, not all the proteases cleave to PAR4 at the same place, and in some cases it results in protection of activation, and in other cases it results to platelet activation. So it's not thrombin doing the job alone in terms of PAR receptor activation, but a bunch of other proteases play a role there as well, and there's also more work needed to exactly find out which of the proteases activate the receptors in which cases. So what's new? On G protein coupled receptor for thrombin, I put ATP in brackets because it has a similar activation pathway. Those roles in thrombosis, hemostasis, and also importantly in thromboinflammation. Relatively low abundance, about 250 copies per platelet, but using pathways of serine threonine kinases like protein kinase C rather than tyrosine kinases, so different initial activation pathway. And there is also another difference is that those receptors, thrombin and ATP receptors, they respond to soluble agonist in a spatial temporal way, meaning that the agonists only are locally active because the agonists like thrombin can be inactivated with antithrombin, ATP will degraded by heparase, so only on the site, site of formation. That is the site, or at least that is the site where those G protein coupled receptors become active, in contrast to the adhesive receptors, which are active for a much longer time. So with it, I come to the end of my presentation. I thank everybody for listening, and